Now, with all things signaling and whatnot, you need to be able to reset. If you can't reset, the signal pathway is only good for one usage. So, evolution has seen fit to evolve us to utilize multiple methods. First method to reset the dominoes is phosphatases, as I mentioned at the beginning of this chapter. The phosphorylated proteins, the phosphorylated receptors, you'll have a protein come along and pluck off those phosphate groups off the tyrosine, the serine, the threonine, whatever. What's going to happen to them? Well, many of the times they're just going to be released. Think about it. Um, they will be released and they will be utilized elsewhere, or they could be reattached to ATPs via substrate level phosphorylation. But anyway, you reset by removing the phosphate groups. Another mechanism is our friend the proteasome. Some of these proteins have been altered once they were activated. So what started off as a much larger protein was cut down into individual polypeptides. Or the phosphorylation of it was just, you know what, it's just too much to go back and reset. So they will be ubiquinated. Ubiquitin is basically a targeted red flag that tells the machinery in the cell to traffic this protein here that's been ubiquinated, traffic it to the nearest proteasome where it'll be broken down into peptide fragments, if not into individual amino acids, which will then be sent back into the amino acid pools to be to recycled. If it's broken down into individual peptide fragments, well, remember, hey, guess what? Some of those can be loaded into MHC1 in the ER using the TAP1, TAP2 proteins to transport it in, and then the whole CLP apparatus to load it into MHC1, which will then take some of those peptide fragments to the surface. Also, what we can see is many of the times these receptors will be tagged, as they're showing here, not quite polyubiquinated, but ubiquinated to the point that the receptor, since it can't be trafficked to a proteasome, well, what you do is you recycle by pulling in that portion of the plasma membrane and sending it to a lysosome. Remember, a lysosome is going to be a vesicle inside the cytoplasm, floating around the cytoplasm, that has lowered pH, digestive enzyme, proteases, RNases, DNases, all the little things you need to break stuff down and recycle the components. Okay, so proteins are great. RAF, RACE, REC, RAF, remember those? RAS, RACE, RAF. Sorry, that's the sequence. Well, we still like these because, like I said, it gives us this amplification process. Instead of one domino knocking over another one knocks over two, those two knock over four. Nobody said that the one knocking over two has to knock over the same two. Well, RAF knocks over a mech. Well, a couple of mechs knock over a couple of ERKs apiece. And notice you started off as one domino, and here you are at four. And those can continue and amplify. But if you think back to 244 intracellular molecular, or 404 if you've had it, proteins, while they diffuse through the cell quickly, it still takes time. So you want something a little bit faster. That's where second messengers come into play. A receptor, G protein coupled receptor, goes through, activates this, that, the other. Certain PIP IP3s get targeted, phosphorylated, PLC comes through, cuts, releases IP3, which is much smaller than a protein, diffuses through the cell very quickly, runs into a gated ion channel, opens up, and then calcium, an individual atom, will flood the cytoplasm within seconds. But we never actually said what the calcium does. Well, one of the things the calcium does is it interacts with proteins that are free floating around in the cytoplasm. One of them is, an example of it is calmodulin. Calmodulin on its own is going to be in an altered shape that has no function. But when calcium binds to certain areas of it, it's going to change the shape. Change the shape, and now giving it a shape that has the function that it can bind an effector protein. Effector protein, this right here is a generic title they're giving this protein. It just means that this is a protein that will do something. But this protein on its own can't do anything. It needs to be bound to calmodulin. Calmodulin is only going to have the correct shape if it's bound to calcium. Calcium's only going to be released from the ER when IP3 opens the gated ion channel. IP3 is only going to be released into the cytoplasm when phospholipase C has been activated. Phospholipase C is only going to be activated when a G protein becomes active. G protein is only going to be active when the receptor is ligated. Oh.